All right. Big thank you to the organizers for the opportunity to present our work, which involves detecting genomic gradients in the human brain. So, the human brain is characterized by a strong relationship between functional specialization and space. So, this relationship can be observed as a modular representation, where brain tissue can be segregated into structures or substructures with shared behavioral functions or cytoarchitecture. And we often call these brain regions. So there's also evidence for gradient-like representations, where gradation in function appears to be organized systematically across specific linear axes. So some examples I'm showing here are gradients in evolutionary organization, neuronal cell distributions, and gradients of cortical connectivity. These regions and gradients coexist and likely interact hierarchically to influence brain functional organization. And that really probably has its origins in gene expression during development. So here are a couple of examples of the developing spinal cord on top and the developing brain at the bottom. In both cases, you can see that gradient-like expression of a few genes help to recruit and coordinate additional gene expression in more discontinuous and sharply bordered patterns, which creates this regional functionalization or specialization. A number of these genes have been identified in the human brain, but it's generally pretty hard to study this phenomenon in humans for obvious reasons. But that's changed somewhat with the introduction of the Allen Human Brain Atlas, which most of you are probably familiar with. It's an open data set. It's an archive of 6 brains from which 3,700 tissue samples have been extracted, each of which underwent microarray gene expression for 59,000 probes targeting 20,000 genes. So a pretty amazing data set. And importantly, each sample also features stereotaxic coordinates, allowing for hypotheses to be tested concerning gene expression in space, which is pretty much what we'll be doing here. So we're trying to understand broadly the relationship between gene expression and space across the whole human brain. And we're asking, are there major spatial gradients along which gene expression systematically covaries? So we'll be using cross-decomposition to try to identify genomic gradients in the adult brain and try to understand relationships between gene expression and the spatial location of a given tissue or cell in the brain. So we start by reducing the dimensions of the genomic data using principal components analysis. We then pass the gene expression data to a cross-decomposition algorithm, such as PLS regression, so as to model gene expression as a function of space. And by space, we literally mean just 3D Euclidean space. So once we fit and validate this model, we can decompose the model into its components, its covariant spatial and genomic gradients. And we can characterize and describe these further, as well as apply them to external data sets, as we'll do here. So we first use a cross-validation framework where we separate tissue samples into training and test sets. We identify the best models for our question at hand using the training set, and then we test the model by predicting the spatial coordinates of left-out tissue samples in the test set using just gene expression information. And if we're satisfied with that step, we can then fit the model to the entire data set, decompose the spatial and genomic gradients, and describe them. So we tested 3 different types of cross-decomposition models, as you can see here. And we tested this with up to 3 components using cross-validation. And we found that the optimal model used partially squares regression and 3 components. And we also saw that all 3 of these PLS components explained variance in the spatial data that was greater than expected by chance by a large degree. And just here, I'm visualizing each of the 3 latent PLS component covariates. So we then used this PLS model to predict the 3D spatial location of left-out tissue samples based just on gene expression. So we found that the model predicted the location very well in these left-out samples along the rostral caudal and the dorsal ventral axes in particular, but less well along the medial lateral axis of the brain, as you can see at the bottom graph there. So to visualize this, here we plot the spatial Y and Z coordinates of each sample in the Allen Brain Atlas, and here's the predicted location of each of those samples from our PLS model. So the predicted version doesn't really look like a brain, but the spatial relationships between the major substructures are more or less preserved, and that's true even if we subdivide this into smaller brain structures. The relative positions are still more or less intact. Here we're visualizing the model error, where red samples, tissue samples, are those that had the most model error. You can see that the model seemed to perform worst in parietal and prefrontal regions. So we were satisfied with the generalizability of the model. So we next fit the model to all the data and we decompose it into its spatial and genomic components so we can sort of describe and evaluate them. So for each one of these components, I'll show you first the genomic gradient at the bottom. So this is the gene expression pattern, and a covariance spatial gradient on top. And so here you can see the first component involves essentially a dorsal to ventral gradient, which separates the cortex from the subcortical structures. And in looking at this, we thought maybe this gradient can differentiate brain regions based on their rostral caudal position in the developing human brain, which is shown here. And as you can see, component 1 expression is lowest in regions that originate in the telencephalon and increases as one moves caudally along the neural tube to regions originating in the met and myelencephalon. So component 2 is characterized by a striking posterior to anterior radiation. And based on our conjecture from component 1, we wondered if this gradient might be important in regulating the dorsoventral axis of the developing brain. So here you can see in each segment of the developing brain, we separated regions based on whether they originated in the dorsal or ventral part of the developing brain, and found that component 2 expression pretty much uniformly was higher in dorsally originating regions, which is pretty interesting. Then if we look just in structures originating in the telencephalon, we find this very specific pattern of component 2 expression that's lowest in the subcortex and increases as one moves from the anterior to posterior parts of the cortex. Finally, component 3 was characterized by a spatial gradient that moves sort of dorsomedially to ventrolaterally. This can probably best be appreciated in the coronal slice on the right. So this genomic gradient obviously less correlated with the spatial gradient compared to the other components, as you can see here, but it did strongly separate association cortex from the rest of the brain. You can perhaps appreciate that better here on this cortical projection, where a primary sensory component, primary sensory cortex has higher component expression in red compared to the heteromodal association cortex in blue. So we were interested in whether these whole brain gradients might intersect and interact to promote cortical arealization. So we basically separated regions into those that shared certain concentrations of these different gradients. We tried this first for the spatial gradients, and that's what I'm showing you here. And if you squint, you might be able to recognize gross anatomical divisions of the cortex. And then we repeated this exercise with the genomic gradients. And this time the gradients defined more of like a sensory fugal axis, where the primary sensory structures, which are here in dark gray, were differentiated from sensory structures, which are in orange, from heteromodal cortex in blue, and limbic structures in green. So next, given the associations we found previously, potential associations with cortical development, we decided to test this more formally and assess when during development these genomic components begin to influence brain spatial hierarchy. So we used the BrainSpan dataset, which is this amazing open access dataset of transcriptomic data for several brains from field development up through adulthood. And this data set includes bulk mRNA transcripts isolated from several structures in the cortex and subcortex, which are delineated here, as well as from the cerebellum. And we're able to apply the weights from our various PLS components that we trained in the Allen Brain Atlas to brain samples in the BrainSpan dataset in order to get component scores in this separate dataset. So recall that the first component differentiated telencephalon originating structures from the deeper, more ventral structures. And I'm just showing you that again here. Now, we replicated the same finding in BrainSpan, where the cerebellar samples had much higher component 1 expression than the cortical structures. And here are the samples. We then visualized how the relationship changed through the course of development. So here you can see the samples arranged from the earliest age, which is 8 post-conception weeks prenatally, to the oldest age, which is 40 years. And we found that the component 1 expression begins to differentiate these structures already during the fetal period of development and plateaus in early childhood. So for component 2, recall that the expression in the cortex differentiated limbic from anterior from posterior structures. And since we were mostly working with cortical structures in BrainSpan, we calculated the mean component 2 expression in several regions in the Allen Brain Atlas and correlated this with the mean component 2 expression of the same regions in BrainSpan. And we found a remarkable correlation between these two data sets, again, indicating generalizability of this component as well. And again, component 2 expression began differentiating structures prenatally. But interestingly, this process seemed to continue through into adulthood. So finally, component 3, as I said before, associated association cortex from sensory cortex, and we were not able to replicate this result in BrainSpan, so we didn't further investigate its evolution through brain development. So in summary, we identified patterns of gene expression that occurred systematically along spatial gradients in the brain. The components were reproducible across and within cohorts and emerged prenatally. And while it requires further follow-up work, these gradients might be involved in cortical arealization. And with that, I'd like to thank all of my co-authors, as well as our funding sources, certainly the Allen Institute and BrainSpan datasets, and thank you for your attention.
Hello and welcome. My name is Philippe Jawinski. I'm a postdoc at Humboldt University, Berlin, and I am very happy having the opportunity to present you with some of our most recent results on the molecular genetics of the biological age of the brain in the UK Biobank. What is aging? For sure, there are many definitions of aging. According to Harman, aging can be seen as the gradual accumulation of deleterious biological changes accompanying a progressive loss of function. So aging increases the risk for morbidity and mortality, and importantly, aging is not experienced at the same rate. So from a biological perspective, you can say that there are individual differences in the speed of aging. So researchers came up with the idea to improve the prediction of age-related disease and mortality by measuring aging from a biological perspective. And this is usually done using supervised machine learning and neuroimaging data. So there's accumulating evidence suggesting an association of the biological age of the brain, of brain age, with several mental health phenotypes. For instance, a recent large study by Kaufman and colleagues showed that brain age is associated with cognitive impairment, dementia, multiple sclerosis, as well as schizophrenia. Now, we wanted to approach the brain aging topic from a molecular genetics perspective. So our aim was to investigate the molecular genetics of brain age and to examine its genetic overlap with mental health traits in the normal and pathological range. So we made use of the most recent release of the UK Biobank sample. We selected 32,000 individuals of white British ancestry with available T1-weighted magnetic resonance imaging data. And all of these samples, they were genotyped. Further, there were imputations which were carried out and to increase the number of genetic variants available for analysis. We just selected those ones which are common, so those ones with a minor allele frequency higher than 1% and with an imputation quality score above 0.8. This resulted in about 9.7 million single nucleotide polymorphisms, as well as insertion deletions suitable for our analysis. And we did different kinds of analysis. We did heritability analysis. We conducted SNP level analysis. Further, we carried out polygenic risk score analysis, as well as so-called genetic effect size distribution analysis to examine the degree of polygenicity. But let me first explain to you how we estimated the biological age of the brain. So we took the T1-weighted images of the UK Biobank subjects and pre-processed them using the CAT12 toolbox for MATLAB. So from this toolbox, we obtained the brain images. They were like transformed from the native space into the standard space. And what we also got was those segmentations into gray matter and white matter brain tissue. So we estimated brain age for both the gray matter and the white matter images. And to further reduce the amount of data, we smoothed and downsampled those images, and we applied a principal component analysis. We decided to take about the first 500 principal components, which accounted for about 90% of the variance occurring in these images here. Now, to build our brain age models, we divided our sample into 10 equal-sized subsets. We took 90% of those 32,000 individuals to train a predictive age estimation model. So we took the 500 principal components as features or predictors, chronological age as outcome variable, and we used different machine learning algorithms. Later on, we just averaged all of the results of those different algorithms used. So with the model built, we estimated the age of the subjects in this test sample here. And so for each of those subjects, you get one brain age estimate, one brain predicted age estimate. And you can repeat this procedure like 9 further times so that you get an estimate of all of the subjects in your sample. These are the results for the gray matter brain age estimates, the white matter brain age estimates, and after combining both the gray and the white matter brain age estimates. What you can see here on the x-axis is the chronological age, and this is the brain predicted age. And you can see that there is a strong correlation between these variables. The correlations were all above 0.8, and the mean absolute error reached about 3.2 years in the combined gray-white matter brain age case. So, but what you can also see is that there is some variance left. So there are some subjects which have who have a higher brain predicted age relative to their chronological age. Some of them have a lower brain predicted age compared to their chronological age. And previous studies have shown that this gap, this discrepancy, is associated with, for example, cognitive performance variables. Now, we wanted to find out whether this discrepancy is also heritable. So we conducted a so-called Gremel analysis. And this genetic restricted maximum likelihood analysis revealed that there is a SNP-based heritability of about 30% for gray matter, 33% heritability for the white matter brain age estimates, and again about 33% for the combined gray and white matter brain age estimates. So this analysis reveals a substantial genetic component for the brain age phenotype, and this is a very, very good prerequisite or very good basis to identify single genetic variants. And this is what we did here. This is a so-called Manhattan plot. Every dot here represents a single genetic variant of the 9.7 million variants that we assessed. They are located on the x-axis according to their genomic position. And the higher a dot here, the stronger the p-value, the stronger the association with brain age. We discovered 12 genome-wide significant findings for the gray matter estimates. And for example, risk alleles of Alzheimer's disease in the ApoE gene, they reached nominally significant levels. So the evidence was even more compelling for the white matter brain age estimates. We discovered 20 independent genome-wide significant findings. And yes, again, risk alleles in the Alzheimer risk allele genes in the ApoE gene were nominally significant. And we have a very, very strong locus here on chromosome 17. Just to give you an idea of the effect size, this variant here accounted for about 0.7% of the variance in brain age, which is a very large effect in the field of complex trait genetics. Again, for the combined gray and white matter brain age estimates, we discovered 19 genome-wide significant findings. And this locus here was again the strongest. So let's have a closer look at this region. This is a so-called regional plot. And what you can see here is the lead SNP for the gray matter brain age results. And there are many, many, many SNPs in high linkage disequilibrium. So they are highly correlated with this lead SNP here, and they span a relatively large genomic region. So it will be probably very hard to pinpoint the causal variant, but there are very plausible genes in these regions here. For instance, the MAP gene, including the lead SNP. This gene encodes the microtubule associated protein tau, and the tau protein has been implicated in several neurodegenerative diseases, with Alzheimer's disease probably being the most popular one. So SNPs in strong linkage have also been found genome-wide significant for other traits, for example, neuroticism, educational attainment, cognitive performance, and so on and so forth. So this is a very, very plausible finding that we have here, and our results, they provide further evidence for a pivotal role of this locus in human complex behavior. Now, what we also did was we tried to replicate all of our discovered variants in some kind of a replication sample, a sample of non-white British ancestry individuals, 5,000 individuals in the sample. And we took the brain age models and just applied them to the T1 magnetic resonance imaging data of this sample. And what you can see is that the prediction performance was comparable to our other sample. So everything well from this point. And what we did now was we took the discovered variants and tested for an association in the replication sample. These are so-called QQ plots. So what we did here was we compared the distribution of observed p-values of the variants tested in the replication sample and compared it to a random p-value distribution under the null hypothesis. And what you can see is that there's a strong inflation, especially for the white matter estimates. So we observed stronger effects than expected by chance, revealing supportive evidence for the discovered variants. To give you some numbers, of the 20 variants discovered for white matter brain age, 18 showed effects in the expected direction, and even 12 of them showed p-values below 0.05, so which is more than expected by chance. All right, what we also did were polygenic risk score analysis. So we took the summary results of previous studies, for example, genome-wide association studies for depression or for cognitive performance. And based on these reported effect sizes, we calculated a genetic risk score for each individual in our sample. You can do this for different p-value thresholds. For example, you can just take variants reported in this study here at the level of genome-wide significance, or you take all the variants showing p-values lower than 0.05, and so on and so forth. So we had 10 different p-value thresholds. And what you can see here are the results of a regression analysis. So these are beta weights. And basically, they can be interpreted as follows. So with one standard deviation increase in the genetic risk score for depression, we see an increase of 0.1 years in gray matter brain age. So these are the gray matter brain age results. In contrast, for example, for cognitive performance, one standard deviation increase in the genetic score for cognitive performance leads to or is associated with a decrease of 0.1 years in gray matter brain age. So these findings are very plausible. And now we did this polygenic risk score analysis for 63 traits of different domains, such as psychiatric traits, but also like risk-taking behavior or personality traits. And all of the colored fields here, they show nominally significant results. So overall, we found that 58 of the tested associations were nominally significant, which is higher than expected by chance. For example, we have seen positive associations with depression, with stroke, with neuroticism. In contrast, negative associations were found for life satisfaction, for positive affect, and for intelligence. Nevertheless, the highest variance explanation that we observed reached about 0.3%. So the predictive accuracy of the polygenic risk scores was very, very low. Surprisingly, for the white matter brain age estimates, associations were somewhat weaker, but still 40% of the associations were nominally significant. And the combined gray and white matter estimates showed results somewhat like in between. All right, one last analysis that we conducted. We wanted to find out whether brain age is a highly polygenic trait. So we conducted a genetic effect size distribution analysis, and we took the polygenic traits neuroticism and height as reference traits. What you can see here is the distribution, the estimated distribution of underlying susceptibility variants for neuroticism. And you can see that there are many, or a relatively large amount of variants with small effect sizes. For height, there is a larger amount of variance, a relatively larger amount of variance with larger effect sizes. And interestingly, for brain age, there are even flatter tails here, indicating that there is a relatively large number of variants, of genetic variants with large effect sizes. So this is also mirrored in those projection analyses here. With increasing sample size, it is expected that the number of genome-wide significant findings drastically increases for brain age relative to the other two traits here. And also with increasing sample size, the amount of genetic variance explained by the genome-wide significant findings is also expected to drastically increase for brain age when compared to, for example, neuroticism. So the bottom line here is that although brain age still is a highly polygenic trait with an estimated number of 5,000 susceptibility variants, when compared to other complex traits, it appears like somewhat, or it appears that the degree of polygenicity is relatively low. So I want to conclude with a SNP-based heritability of about 30%. Brain age constitutes a trait with a strong genetic component. We discovered 29 independent genomic variants, of which a considerable proportion appears to replicate across populations. Polygenic risk score analysis suggests an involvement of brain age in a variety of mental health traits in the normal and pathological range. When compared to other complex traits, brain age shows a relatively low degree of polygenicity. And what we now need are Mendelian randomization analysis to find out or to clarify the role of brain age as potential cause or consequence of health phenotypes. So these are my references. I want to thank the whole molecular psychology team for their contributions, especially Sebastian Marquette and Helena Braun. And of course, I also want to thank you for your attention. Stay safe and take care.
We're fortunate to be researchers at a time when we have access to a wider range of data than ever before. And this is due to the increasing size and number of publicly available data sets, as well as any data that is collected in-house. But this has allowed us to develop increasingly more complex research questions and statistical tests to answer those questions. So one area our lab is particularly interested in is using this complex multivariate data to and synthesizing it to identify one's clinical trajectory. However, this increased complexity has also uncovered novel issues to be considered, such as the high heterogeneity that exists between individuals. So for example, when considering the total aging population, you have typical agers who age exactly basically as we might expect. You also have resilient agers who age perhaps more efficiently than you would expect, and they typically have younger looking brains and better cognition than expected. And then you also have advanced agers. Now, it should be clarified that all of these fall within the grouping of cognitively normal, but even within that categorization, we have this heterogeneity. And then we believe if you further investigate the advanced group, you'll see that they will probably follow one of several trajectories. So not everyone is advanced in the same way. It's likely that one or more of these trajectories is associated with pathology related to diseases such as Alzheimer's, so that if you identify one's trajectory, you might also have a better understanding of their future outcome. So the idea behind this study is to combine a large sample with complex multivariate data and machine learning to identify individualized biomarkers. And then this will help us to more effectively meet our goal of identifying one's clinical trajectory. To do this, we have worked with many others to develop the iStaging Consortium. The iStaging Consortium consists of multiple data sets, which you see here, and they have varying scope, but really they generally focus on aging. And they include data from each of the modalities mentioned previously, so MRI, PET, and also the clinical and cognitive domains. Currently, there are around 30,000 participants, some with longitudinal time data, so there are many more time points actually than you see here. In general, the majority of these are cognitively normal, but there also are some patients with mild cognitive impairment or Alzheimer's disease. Importantly, our lab has worked to harmonize these data by site so that we can work with the data as if it comes from a common source.
Hi everyone, my name is Jingwei. I'm currently working in the Jülich Research Center as a postdoc. Today, I will talk about the different performance of resting state functional connectivity-based behavior prediction models in African American and White American samples. This work is done jointly in the Jülich Research Center and National University of Singapore. Recently, there has been increasing research interest in relating resting state functional connectivity data to behavior measures. These studies focus on the association between inter-subject variation in functional connectivity and the individual differences in behaviors. Usually, this relationship is studied on the whole population. However, in the machine learning research area, people start to care about the fairness of the machine learning algorithms across different subpopulations. Here is a simple example. A study in 2019 found that when commercial gender classification systems were trained on a dataset with more lighter-skinned subjects, the darker-skinned female samples suffered higher misclassification rate. There could be many reasons leading to the unfairness of the algorithms. Firstly, it is possible that the model can't learn enough information for the minor group due to the limited sample size. The second reason could be, since the data is dominated by majority population, the algorithm will put more efforts on reducing prediction error of the majority to reach lower values of cost function. And there can be other reasons as well. Hence, in our research area, it is important to explore the question whether the behavior prediction model trained on whole population performs equally well in different subpopulations. Here, we studied that for different reasons. We used the Human Connectome Project data. It contains a large range of behaviors, from where we selected 58 behaviors across cognition, personality, and emotion. It also contains multiple races. We used 948 subjects with 700 white Americans and more than 100 African Americans. We also considered some covariates like age, sex, motion, education, household income, and intracranial volume. The resting state fMRI data were preprocessed by IC-FIX with global signal regression. First, we need to make sure that the African American and white American samples are comparable, so we need to match their distributions of covariate and behavioral variables as much as we can. The following process was repeated for each behavior multiple times to find at least 40 random ways to split the subjects into matched folds. First, we split the African Americans randomly into 10 folds, and the subjects in the same family did not appear in different folds. Then we also randomly split the same number of white Americans and did Hungarian matching with African American folds based on age, sex, motion, and behavior scores. After 10,000 iterations, we selected the iteration with lowest matching cost. Note that we also try to match for education, household income, and intracranial volume, but these measures are impossible to be matched unless we throw a large portion of African Americans. We also found that there were 7 behaviors can't find good match between white Americans and African Americans. They won't be included in the following analysis. For the remaining behaviors, the matched white Americans and African Americans showed no significant difference with respect to age, sex, motion, and behavior scores. Then we split the remaining white Americans, 28 outlier African Americans, and the subjects with other races randomly into 10 folds. Then for each fold, the 3 groups of subjects were combined. We have ensured that there was no family overlap across the final folds. We used clonal regression models to predict behaviors. This method is intuitive and has been proved to achieve relatively good prediction accuracies. It predicts the behavioral score of a test subject as the weighted average of the behavioral scores of all training subjects, and the weights are determined based on the similarity of functional activity across subjects. Here we used functional connectivity computed across 400 cortical RIs and 19 subcortical RIs. We performed 10-fold cross-validation, and we explored the test accuracy between African Americans and white Americans when age, sex, motion, education, income, and ICV were regressed from either behaviors or functional connectivity. For each case, the accuracy differences were tested by randomly permute the race of subjects for each behavior, and FDR correction was done for multiple comparisons. We used 3 accuracy metrics. Taking African Americans as an example, Predictive COD is 1 minus the fraction between residual sum squares of African Americans and the total variance of both African Americans and white Americans in the training folds. It can be interpreted as how much of the total variance can be explained by the model in African American samples. The denominator is defined in this way because it assumes that the total variance should not change by knowing the sensitivity attribute. We computed Pearson's correlation for either race group. We also defined the normalized MSE for each race group. It is defined as the regional MSE divided by the variance per race group in the training sample. This metric considers the difference of true behavior score between African Americans and white Americans. Next, I will show you the results using predictive COD as the accuracy metric. When regressing the confounding variables from behaviors, 29 behaviors can be successfully predicted. Here, predictable means it survived the permutation test by shuffling the predicted score across subjects, and the predictive COD value is positive in either African Americans or white Americans. There are 26 behaviors showed significant accuracy difference between the two groups. White Americans had higher predictive COD in all these 26 behaviors. Similarly, when the regressors were regressed from functional connectivity, we still found that most behaviors showed significantly higher predictive COD in white Americans than African Americans. When using correlation as the accuracy metric, the predictable behaviors were roughly the same as using predictive COD, and most of them exhibited significant differences between the two groups. What to be noted is now we found some behaviors where the correlation accuracy of African Americans is higher than white Americans. In this figure, I will show you the correlation accuracy for each individual behavior. Blue represents African Americans, green color corresponds to white Americans, and red is green minus blue. The star means the difference is significant with permutation test after FDR correction. The behaviors are sorted by the difference between two groups. Behaviors on the left had higher correlation in white Americans, and behaviors on the right had higher correlation in African Americans. These are the behaviors showed inconsistency between predictive COD and correlation metrics, which need further exploration. The first reason could be that correlation can't capture the overall shifts of prediction. Here I show an exemplar behaviors scatter plot. X-axis is the true behavior score, and Y-axis is the predicted score. Each blue dot represents one African American subject, and green dots are white American subjects. Visually, African Americans do have more positive slope, but numerically their predictions deviate more from the average true scores. The second reason could be the variance of African Americans' true behavior score is higher than white Americans, so that the sum square of residuals tend to be higher for African Americans, leading to lower predictive COD. In this example, the overall shift of the predicted score for African Americans is lower than white Americans. But the variance of raw behavior scores for African American is greater than white Americans. In this case, difference in predictive COD could be driven by difference in raw score variance. Generally, we observed higher raw score variance and higher prediction shift in African Americans, even for the behaviors with consistent conclusions using predictive COD and correlation. But for those consistent behaviors, the larger prediction error in African Americans caused both lower predictive COD and lower correlation accuracy. Then we try using MSE normalized by each race group's variance of behavior scores. Here we consider the same behaviors as that were predictable using predictive COD. There were fewer behaviors with significant difference in normalized MSE. Here I will show you the normalized MSE for individual behaviors. Same as before, blue means African Americans, green means white Americans, and red is the difference. Star indicates the difference is significant. We can see that the red bars are more overlapped with the zero line, implying that Part of the group difference in predictive COD was related to different variants. Here are some take-home messages. First, a perfect matching between African Americans and white Americans can't be achieved for some variables using current data. What we can do for now is to regress them from either behaviors or functional connectivity. But simply regressing those variables does not really help to fill the gap. It is possible that there are some other variables we haven't considered. It could also be that the influence of these variables is not linear. We also observed that African Americans tended to have higher behavioral variance in our current samples. Like what is shown in this histogram, African Americans' behavioral scores spread more to the tails, and the group difference in behavioral variance can affect the prediction accuracy metrics. To better study the performance of behavior prediction models in different subpopulations, we need better matching between the subpopulations. Hence, more data for the minorities need to be collected. We'd like to explore this question using other datasets like UK Biobank or NKI, but we're still concerned that the data for minorities are not enough. For example, in the UK Biobank data, the second largest ethnic group is Asian British, but it only occupies 1% of the total sample size. There are only around 300 Asian subjects with both functional connectivity and cognitive behavior data collected before any quality control. So we need some databases which have larger representation of minorities. I would like to thank Sarah, Thomas, Danilo, and Avram for their insightful comments. I also want to thank the funding resources. Thanks for your attention. Hi, everyone. My name is Jingwei. I'm currently working in the Ulrich Research Center as a postdoc. Today, I will talk about the different performance of resting state functional connectivity-based behavior prediction models in African American and white American samples. This work is done jointly in the Ulrich Research Center and National University of Singapore. Recently, there has been increasing research interest in relating resting state functional connectivity data to behavior measures. These studies focus on the association between inter-subject variation in functional connectivity and the individual differences in behaviors. Usually, this relationship is studied on the whole population. However, in the machine learning research area, people start to care about the fairness of the machine learning algorithms across different subpopulations. Here is a simple example. A study in 2019 found that when commercial gender classification systems were trained on a dataset with more lighter-skinned subjects, the darker-skinned female samples suffered higher misclassification rate. There could be many reasons leading to the unfairness of the algorithms. Firstly, it is possible that the model can't learn enough information for the minor group due to the limited sample size. The second reason could be, since the data is dominated by majority population, the algorithm will put more efforts on reducing prediction error of the majority to reach lower values of cost function. And there can be other reasons as well. Hence, in our research area, it is important to explore the question whether the behavior prediction model trained on whole population performs equally well in different subpopulations. Here we studied that for different races. We used the Human Connectome Project data. It contains a large range of behaviors, from where we selected 58 behaviors across cognition, personality, and emotion. It also contains multiple races. We used 948 subjects, with 700 white Americans and more than 100 African Americans. We also considered some covariates like age, sex, motion, education, household income, and intracranial volume. The resting state fMRI data were preprocessed by IC-FIX with global signal regression. First, we need to make sure that the African American and white American samples are comparable, so we need to match their distributions of covariate and behavioral variables as much as we can. The following process was repeated for each behavior multiple times to find at least 40 random ways to split the subjects into matched folds. First, we split the African Americans randomly into 10 folds, and the subjects in the same family did not appear in different folds. Then we also randomly split the same number of white Americans and did Hungarian matching with African American folks based on age, sex, motion, and behavior scores. After 10,000 iterations, we selected the iteration with lowest matching cost. Note that we also try to match for education, household income, and intracranial volume, but these measures are impossible to be matched unless we throw a large portion of African Americans. We also found that there were 7 behaviors can't find good match between white Americans and African Americans. They won't be included in the following analysis. For the remaining behaviors, the matched white Americans and African Americans showed no significant difference with respect to age, sex, motion, and behavior scores. Then we split the remaining white Americans, 28 outlier African Americans, and the subjects with other races randomly into 10 folds. Then for each fold, the 3 groups of subjects were combined. We have ensured that there was no family overlap across the final folds. We used clonal regression models to predict behaviors. This method is intuitive and has been proved to achieve relatively good prediction accuracies. It predicts the behavior score of a test subject as the weighted average of the behavior scores of all training subjects, and the weights are determined based on the similarity of functional activity across subjects. Here we used functional connectivity computed across 400 cortical RIs and 19 subcortical RIs. We performed 10-fold cross-validation, and we explored the test accuracy between African Americans and white Americans when age, sex, motion, education, income, and ICV were regressed from either behaviors or functional connectivity. For each case, the accuracy differences were tested by randomly permute the race of subjects for each behavior, and FDR correction was done for multiple comparisons. We used 3 accuracy metrics. Taking African Americans as an example, predictive COD is 1 minus the fraction between residual sum squares of African Americans and the total variance of both African Americans and white Americans in the training folds. It can be interpreted as how much of the total variance can be explained by the model in African American samples. The denominator is defined in this way because it assumes that the total variance should not change by knowing the sensitive attribute. We computed Pearson's correlation for either race group. We also defined the normalized MSE for each race group. It is defined as the regional MSE divided by the variance per race group in the training sample. This metric considers the difference of true behavior score between African Americans and white Americans. Next, I will show you the results using predictive COD as the accuracy metric. When regressing the confounding variables from behaviors, 29 behaviors can be successfully predicted. Here, predictable means it survived the permutation test by shuffling the predicted score across subjects, and the predictive COD value is positive in either African Americans or white Americans. There are 26 behaviors showed significant accuracy difference between the 2 groups. White Americans had higher predictive COD in all these 26 behaviors. Similarly, when the regressors were regressed from functional connectivity, we still found that most behaviors showed significantly higher predictive COD in white Americans than African Americans. When using correlation as the accuracy metric, the predictable behaviors were roughly the same as using predictive COD, and most of them exhibited significant differences between the 2 groups. What to be noted is now we found some behaviors where the correlation accuracy of African Americans is higher than white Americans. In this figure, I will show you the correlation accuracy for each individual behavior. Blue represents African Americans, green color corresponds to white Americans, and red is green minus blue. The star means the difference is significant with permutation test after FDR correction. The behaviors are sorted by the difference between 2 groups. Behaviors on the left had higher correlation in white Americans, and behaviors on the right had higher correlation in African Americans. these are the behaviors shows inconsistency between predictive COD and correlation metrics, which need further exploration. The first reason could be that correlation can't capture the overall shifts of prediction. Here I show an exemplar behaviors scatter plot. X-axis is the true behavior score, and y-axis is the predicted score. Each blue dot represents one African American subject, and green dots are white American subjects. Visually, African Americans do have more positive slope, but numerically, their predictions deviate more from the average true scores. The second reason could be the variance of African Americans' true behavior score is higher than white Americans, so that the sum square of residuals tend to be higher for African Americans, leading to lower predictive COD. In this example, the overall shift of the predicted score for African Americans is lower than white Americans. But the variance of raw behavior scores for African American is greater than white Americans. In this case, difference in predictive COD could be driven by difference in raw score variance. Generally, we observed higher raw score variance and higher prediction shift in African Americans, even for the behaviors with consistent conclusions using predictive COD and correlation. But for those consistent behaviors, the larger prediction error in African Americans caused both lower predictive COD and lower correlation accuracy. Then we try using MSE normalized by each race group's variance of behavior scores. Here we consider the same behaviors as that were predictable using predictive COD. There were fewer behaviors with significant difference in normalized MSE. Here I will show you the normalized MSE for individual behaviors. Same as before, blue means African Americans, green means white Americans, and red is the difference. Star indicates the difference is significant. We can see that the red bars are more overlapped with the zero line, implying that part of the group difference in predictive COD was related to different variants. Here are some take-home messages. First, a perfect matching between African Americans and white Americans can't be achieved for some variables using current data. What we can do for now is to regress them from either behaviors or functional connectivity. But simply regressing those variables does not really help to fill the gap. It is possible that there are some other variables we haven't considered. It could also be that the influence of these variables is not linear. We also observed that African Americans tended to have higher behavioral variance in our current samples. Like what is shown in this histogram, African Americans' behavioral scores spread more to the tails, and the group difference in behavioral variance can affect the prediction accuracy metrics. To better study the performance of behavior prediction models in different subpopulations, we need better matching between the subpopulations. Hence, more data for the minorities need to be collected. We'd like to explore this question using other datasets like UK Biobank or NKI, but we're still concerned that the data for minorities are not enough. For example, in the UK Biobank data, the second largest ethnic group is Asian British, but it only occupies 1% of the total sample size. There are only around 300 Asian subjects with both functional connectivity and cognitive behavior data collected before any quality control. So we need some databases which have larger representation of minorities. I would like to thank Sarah, Thomas, Danilo, and Avram for their insightful comments. I also want to thank the funding resources. Thanks for attention.
So thank you for attending my talk. Today, what I'm going to talk about is toward the connectivity gradient-based framework for reproducible biomarker discovery. I'm going to give a short background. So as you know well, this network neuroimaging research now has a relatively long history, like 2 decades. And then initial focus of the research was targeting specific connection or networks of interest, such as DMN or frontopedia network. Now it has been increasingly toward more a system-level principle of large-scale network organization in order to understand overarching principle of functional brain.
which is really important and interesting question, but the approach has been so far substantially hampered by ultra high dimensionality of whole brain connectivity data. That's why back in like late 1990 or even early 2000, those pioneering researchers or groups tried to apply this low dimensional representation by performing dimensionality reduction techniques to the resting state fMRI, for instance. Each of which this study make the clear point to sell the utility of this dimensional reduction technique, but only recently after this paper came out, this situating the deformant network along a principal gradient of macroscale cortical organization, the advantage of this dimensional reduction technique to the resting state fMRI has been more exposed to the public, because it not just sticking on to reducing the dimensionality per se, but it turns out that this functional gradient can recapitulate important neurocognitive axis that is already proposed by other pioneering researchers such as Master in 1998. So after this paper, many recent papers start to use this technique in both clinical neuroscience and cognitive neuroscience, so that we thought that this is really the right time for us to check the integrity of these functional gradient features. So recently, we made the bioarchive study where we assessed key biomarker trace of functional gradient with respect to reproducibility, reliability, and predictive validity. And the predictive validity means how much the gradient feature could predict the independent phenotypic data. And then while assessing this trait, we tested following different conditions. The first one was the dimensional reduction algorithm, either linear or nonlinear method, which one give a higher reliability or reproducibility. We also checked the input data representation, either a raw time series, unthreshold or differently threshold functional connectivity, again, which one give a higher reliability. And lastly, we test this feature as a function of amount of the data from 5 to 15 minutes, so that you can see the acceptable data quality can be from how long this functional MRI data. So by performing this, we use these two independent samples. One is Human Connectome Project, and the other one is Midnight Scan Club for the finding of discovery and replication. So, and then we use this minimally preprocessed resting state fMRI time series and connectivity. So from now on, I'm going to show you the result of reproducibility and then reliability and predictive validity in order. And this is first slide. So as you see, this is raw transparently shown the gradient profile across different algorithms. PCA is representative linear dimensional reduction algorithm. Diffusion embedding and Laplacian Eigen maps are also famous popular algorithm for nonlinear dimensional reduction algorithm. And as you see here, although you have to take into account the flip of a sign, the relative patterns are, gradient patterns are similar up to 6's gradient. And then from 6, they start to show algorithm specific patterns. And then now if you see this graph, regardless of algorithm, if you increase the threshold of functional connectivity, for instance, from 0, 25, 50, until 90% of threshold, you can see this curve is get flattened out, which basically show the variance explained, which means that if you increase the threshold, the variance explained by each gradient component is less dominated by first few gradients. The other finding is now we compare the gradient profile between these different algorithms, and then before we align the gradient to the group template, it seems that those algorithms give somewhat different profile, but after alignment to the group template, you see the similarity get recovered from here. So that it turns out that this alignment process is really important before performing any analysis. And again, when we check the discovery and replication reproducibility, it turns out that 3 algorithms show or high reproducibility. Now this is slide for reliability. Here we use the intra-class correlation coefficient and then discriminability for the reliability measures. And if you focus on these first 4 graphs, ICC, so y-axis ICC, and then it generally tends to decrease as a function of the gradient order, regardless of algorithm. And then regardless of algorithm, actually, if you see now the color transition from the red to blue, it is actually unthreshold, less threshold, and more and more highly threshold. It actually shows higher reliability. So it suggests that we have to perform some kind of thresholding process. Now, if you see the ICC guideline, this is somewhat established guideline. According to this, now if you see this inset graph here, from 95 to 97% of threshold shows acceptable reliability across 3 algorithms. And then again, compared to the other nonlinear dimensional reduction algorithm, PCA turns out showing highest ICC, as you see also in this whole brain map. So this is 10 gradient, and then PCA shows highest ICC relative to other algorithms. What is interesting is that now if we stratify this ICC map with respect to this established Yeo-Crane and Atlas, it shows that those high-order transmodal system that include DMN and frontopediatal network shows highest ICC. This is another reliability test as a function of amount of the data. Obviously, if you have more and more larger amount of the data, then you could have higher reliability. Now, if you see, if you compare these 3 algorithms, PCA always show higher ICC. And if you see now this graph, from 10 minutes to 20 minutes, 20 minutes start to show a little bit of excellent ICC. So if your data acquisition status cannot really afford more than 20 minutes, at least you are better to have, we may be better to acquire 20-minute longer resting static MRI to obtain the acceptable reliability. Now, if it's this graph, regardless of which brain area or network systems are, as a function of amount of the data, it is monotonously increased. So there is no really differential pattern across the brain areas. Now, finally, we check the predictive validity. And I'm not gonna go the details. If you want to know the details of our prediction framework, you can refer to the bioRxiv paper, which I show you in the first slides. But then briefly, we use this canonical correlation analysis, and the task was using 1 gradient to predict 1 phenotypic score out of 65 different phenotypic scores that HCP Human Connectome Project provide us. And now if you see this result, this is the result of prediction accuracy. And y-axis is 100 different gradient, and x-axis 65 different phenotypic scores. And each cell here, like a lot of cells you can see there, each cell represent the prediction accuracy. And if you see the yellow or red color, it means they are all well predict. If you see the blue, either blue or green color, it suggests that it's not so predict well. So as you see, out of this is typo, I'm sorry, so 65 phenotypic scores. As you see, the cognition domains are highly predictable compared to the other. Psychiatric and life function also little bit predicted, but not emotion and sensory. What is really main finding in my study was now we correlated this whole brain ICC and prediction accuracy. It shows strong positive relationship, which suggests that if your measure is highly reliable, there is high hope that your measure can predict unseen phenotypic scores. So basically, you better to have reliable measures. So this is another predictability test. Now you can compare between different algorithms, PCA, diffusion embedding, and Laplace Ionizer map. And as you see clearly, the PCA always shows higher prediction accuracy. For instance, those low threshold functional connectivity didn't really show any prediction accuracy in both diffusion embedding Laplace Ionizer maps. Another interesting part is that now we check which brain area is actually more predictable, and it turns out that those again high order transmodal system like frontoparietal network and DMN turns out to be highly predictable compared to other brain areas. Finally, we also check as a function of the amount of the data. Obviously, if you increase the amount of the data, the prediction accuracy get increased. So again, if you cannot afford more than 30 minutes, at least try to have 20 minutes of resting state fMRI. Finally, we compare the prediction accuracy of gradient approach to a reference method. Here we select the connectome-based predictive modeling as a reference, which was developed by Todd Constable Group. This is really simple but powerful tool. It showed that actually our multivariate functional gradient has higher prediction accuracy, both in externalized symptom and general cognition. And if we combine multiple gradient, we can even increase the prediction accuracy. So we perform this biomarker test with respect to reproducibility, reliability, and predictive power according to different conditions. And here, finally, I want to make important point with respect to analytical strategies. So please match the gradient orders and sign before any analysis. Use linear dimensional reduction algorithm like PCA, and utilize the gradient that explain greater amount of variance of the original data. Try to extract gradient using more conservatively threshold functional connectivity, like 95, 96% of threshold. And finally, try to focus on more reliable and predictably powerful high-order transmodal system rather than low-level primary sensory system. So this is my end, and these are my colleagues, and thanks for Mike Milham and Adreda Di Martino. And importantly, I recently joined the university in Korea. So if you have interest in joining our lab, please come to our website and see the job posting. Thanks.
Okay, I am Bertha, and I am here to talk to you about some results on how brain communication is shaped by cortical heritage. Based on previous research, it is known that the myelin content, the cortical thickness, and many other structural and functional properties change across the brain from sensory motor regions towards higher order areas. We can derive these same patterns from connectivity data when we explore the similarity between regions according to their connections to the rest of the cortex. So, using a decomposition of the functional connectivity data, we can organize the brain areas into a spectrum that hierarchize the brain from unimodal to transmodal regions. The question is now, what are the effects of this heterogeneity in the communication? To answer this, we establish signal trajectories and then study how the signal flows within the hierarchical organization of the cortex. From diffusion spectrum imaging and deterministic streamline tractography, which quantifies the number of anatomical fibers connecting two brain regions, we derive the structural connectivity. From this connectivity matrix, we compute the weighted shortest paths between all pair of nodes. From fMRI, we quantify the temporal correlations in spontaneous activity between brain regions, and derive from there the functional connectivity. Then, using diffusion map embedding, we got the gradient that hierarchizes the brain along a continuous gradient from unimodal to transmodal regions. We label nodes according to the hierarchical position, and in this way we get paths through the unimodal transmodal hierarchy. Using the cumulative distribution of the gradient, we bin nodes into 10 groups of equal size. We enumerate the classes from 1 to 10, where 1 corresponds to unimodal and 10 to transmodal. For a source target class, we consider all the possible paths between the constituent source target nodes and average the hierarchical position of every step along the paths, separating the paths by their length. Each color here corresponds to a different length. We can see how the path motif shape depends on the hierarchy of the source and target. They follow the hierarchy, but there are some systematic differences. So we investigate how the orientation of the path changes along the trajectory, both at a nodal level. For a given node, we calculate the average slope of all the paths as they pass through that node. If the average is positive, it means that the transmission is in general to higher hierarchies, while a negative average means that the information is being directed to lower hierarchies. We assume that the hierarchical position of a node is negatively correlated with its average slope. Nodes that occupy lower positions in the hierarchy tend to direct the signals towards nodes higher in the hierarchy and vice versa. When we plot these over the brain, we can see areas like the prefrontal, the auditory, the intertemporal cortex showing a negative slope, while areas like the supplementary motor area, the somatomotor, or the visual cortex have a positive slope. When we group these nodes, but by resting state networks, we see that the visual, the somatomotor, the frontoparietal, the ventral attention, the dorsal attention, all those networks have a mean positive slope, and the limbic and the default mode network have a mean negative slope. The default mode network appears to be the most likely to be sending the information to areas lower in the hierarchy, while the visual, the ventral attention, the dorsal attention are sending information to areas higher in the hierarchy. As we follow a path, we can see that there are some points where the trajectory is redirected. What I mean here is there are points where the slope changes in the time. We call these points turning points, and we name them turning up if the change in the slope is from a negative to a positive slope, or turning down if the change in the slope is from a positive to a negative slope. To give an example, a turning down point is a node that occupies a relatively higher position in the hierarchy and connects two lower level areas. We measure the probability of a node to act as a turning point and found that the regions like the superior and the dorsolateral prefrontal, the intertemporal and the lateral temporal cortex have the greatest probability of turning down the paths, while areas like the supplementary motor area and the posterior parietal cortex have the higher probability of turning up the paths. This behavior becomes clearer when we group the nodes by resting state networks. We find that the attention-related networks have the highest probability of turning up the paths, whereas the default mode networks has the greatest turning down probability. From this, we learn that attention-related networks are redirecting signal traffic up, and default mode networks nodes are redirecting the communication paths down. This result agrees with some theories that identify attention and control networks as the bridges between segregation and integration of information. To summarize, we ask, what are the effects of the hierarchical organization of the brain over the communication? So, we studied what sequences of hierarchies were visited along the shortest path and found, first, that the architecture of the network promotes signaling via the hierarchy, and the cortical hierarchy shapes the communication motifs. This suggests a link between structure and function on the network. Second, the average slope and the hierarchical position of a node are negatively correlated. So, the transmodal nodes direct information to lower hierarchies, and the unimodal nodes direct information to higher hierarchies. Third, the cortical hierarchy shapes the communication paths, even redirecting the slope. This means that the default mode network appear to be most likely to send information to areas lower in the hierarchy, while the dorsal attention and ventral attention are sending information to areas higher in the hierarchy. There are more interesting results in the paper that we published with this work. For example, there is a memory effect in the shape of communication patterns, such that the position of the signal depends on the previous position visited. So I really invite you to read the paper. The results that I presented here open new questions, like what are the transformations that a signal experiences while propagating through a heterogeneous brain? Or what will be the influence of the hierarchy over the communication processes, different ones like navigation or diffusion? Answering these questions could shed more light on the role of the spatial gradients and its interaction with macroscale network topology. Finally, but not less important, I want to recognize the help and support of the amazing lab I am part of. And thank you for listening.